How can we go from spiking neurons to rate neurons? Welcome to this video. My name is Wolfram Gerstner. I would like to present the paper of Valentin Schmutz, and I would like to discuss how rate-based dynamics can emerge in spiking neural networks. Let me start with the problem of finding rates. A standard way to calculate a rate is by averaging over time. Suppose I present a stimulus to a brain. I record in visual cortex V1, I record a single neuron in a single trial. If the stimulus is presented for a long time, say 1 second, the neuron will respond with many spikes. I can count the number of spikes, divide by the big T, and this gives me a finding rate. So this rate is a single neuron rate by averaging over time. Now there's a different way of calculating rates, and the different rate is important because averaging over time is problematic. Suppose that the stimulus is not 1 second, but is only 200 milliseconds. Then my neuron will only give maybe a single spike or maybe 2. So averaging no longer makes sense. Temporal averaging no longer makes sense. Instead, what we can do is we average over a population of neurons. You can think of this as a spatial average, but it's really an average over a population. So suppose I record in visual cortex V1, I present my stimulus, and I record from many neurons at the same time. So neuron 1 emits some spikes, neuron 2 emits some spike, neuron 3 emits some spike. Different neurons emit spikes. What I do, I count spikes over the neurons in a very short time interval, and this way I get a time-resolved measure. It's a single trial, it's multiple neurons, and essentially it's an average over a population of neurons. And ideally this population of neurons should consider of cells that have similar properties. For example, we take layer 5B pyramidal cells in visual cortex V1. So with this concept in mind, we count the spikes across the neuron, we divide by the number of neurons in our recording, and we divide by delta t, and this gives a quantity which has units of rates, and it's the population activity or population rate. Suppose this is now my rate across a population of similar neurons. Then in the brain I will have not just one of these populations, I have many of these. These different populations talk to each other. Say this is the activity in my population N. It's sent over to other populations, and from there yet to other populations and back. So I have now a recurrent network of connected populations of neurons. Let's take the point of view of a single population, AN. It receives input from this population and from that population and from this population. So different populations talk to each other, and they talk to each other in terms of rates. So now we can think of a rate network where each rate neuron is, or each rate unit, I should say, is a population of neurons. This notion of population activity makes a lot of sense if I take the point of view of a postsynaptic neuron K, a single neuron. This neuron has many synapses, thousands of synapses. So in a certain sense, it already takes the average over incoming spikes. And now, if these incoming spikes come from two different populations, a black one and a gray one, then I could say this neuron sees the population activity of population A1 and population A2. However, for this population averaging, we had this notion of averaging over a population of neurons that consists of similar neurons. Then the question is, are these presynaptic pools really homogeneous enough that I can think of these populations as containing similar neurons, or ideally duplicate neurons, neurons of the same type, of the same properties? And when I talk to my experimental colleagues, they tell me, no, it's a rather homogeneous group, and these synaptic rates give a weighted average over a heterogeneous group of inputs. So we have two standard definitions of rate codes. Both are averaging methods. One is an average over time. It's too slow for the animal. It's too slow if the stimuli change rapidly. And the other one is the average over population. Intuitively, it requires averaging over a homogeneous group of neurons. And then the question is, well, do we really have these duplicate neurons defined as neurons with very similar properties? And so this is the big question of this video. Is a rate description meaningful in spiking neurons if temporal averaging is not possible because signals are fast, input changes rapidly, and spatial averaging or population averaging is not possible because there are no duplicate neurons? And now you kind of ask yourself, how should I answer to this question? Because in a way, it's intuitively plausible that it should be possible neurons take averages, but then it's not plausible because homogeneous populations don't exist in the brain, and maybe there's a way out, but then what are the conditions? So we want to go from spiking neurons to rate neurons, and we want to do this without duplicates. And this is what I'm going to show in the remainder of this video. Let me review the notion of functional similarity. So we know that neurons have receptive fields. For example, neurons in the head direction system respond to the direction of heading or the direction of gaze of the animal. And if I apply a first stimulus here, then the yellow neuron would be active. That's the neuron that I record here. If I apply a stimulus here, then the blue and the green neuron would be active. And these neurons don't need to be located to each other. What matters is the fact that they respond similarly. Therefore, I can resort my neurons based on similarity, and then the blue and green neuron are next to each other on this circle where the position is a variable z, and this is the position on this ring. And that gives a ring model. Neighboring neurons have similar responses, and when I say neighboring neurons, I don't mean in the cortical space. Functionally similar neurons do not have to sit next to each other in cortex. So here's the ring model, and what I plot here are the correlations between two arbitrary neurons of my ring model. And suppose here's again on the right hand side I have my head direction alpha. I give a stimulus here, and I have many different neurons that all have their preferred head direction. And then if the stimulus is this stimulus here, then this group of neurons would show correlated activity. Now I change the stimulus randomly and I put it here. Now it's a different group of neurons. I change it again, and now it's a different group of neurons that respond. Therefore, there are many pairs of neurons, ij, that are correlated. And in fact, there are many neurons that show high correlation close to 1. And of course, these high correlations are caused by pairs of neighboring neurons. So now we define duplicate neurons as neurons that are either strictly identical or strongly correlated. And so we see in the ring model, many pairs of neurons are strongly correlated. Therefore, the ring model has duplicate neurons. And duplicate neurons, in that case, means they respond nearly the same. So that was the ring model. Now let's look at a different model, a model that's called a low-rank model. The low-rank model is given by a connectivity matrix Wij, which I can define as a sum over random numbers Xi mu, Xj mu, and the sum ends at the value p. That's the low number p that gives the low rank connectivity. And now let's consider not the Hopfield type where the Xi mu would be binary, but in our case the Xi mu or Xj mu are Gaussian distributed. And then I give different stimuli that change over time, and I ask, how is the correlation between different pairs of neurons? And now if I have the p equal 2, then indeed there are again pairs of neurons that are correlated. If I choose p equal 4, there are already much fewer neurons, pairs of neurons that are correlated. And if I take p equal 50, then it may look like this, and there are hardly any neurons that are correlated at a value close to 1. So this was for a fixed number of neurons in my network. I can do something similar and say I have a number of patterns p, this low rank number, that is proportional to n. And now if n is 1,000,000 as before, and if then I have p equal 100 patterns, or low rank with p equal 100, and then you see there are no correlated neurons left. Now if I have only 10 to the 5 neurons, then, well, the curve looks like this, and there are still a little bit of correlated neurons left. So what we have is neurons become uncorrelated if n is big, p is also big, and the ratio goes to 0. And the simple way of doing this is, for example, that we take p equal n to the power of 1/3, and so this corresponds to p equal 100 in 1,000,000 neurons. And then in this series there's never any duplicate neuron. So this is our first important finding for a network with a low rank weight matrix, which means that the number of terms is restricted to p, which is the number of patterns, and the patterns here are Gaussian distributed. The neurons in such a network will be uncorrelated, and there are many different ways to get uncorrelated neurons. The first one is we have p equal alpha n, p is proportional to n, and then we let go n to infinity, neurons become uncorrelated. Or we can also go take p to infinity for fixed n. But most interesting for the following is the other option. P goes to infinity, n goes to infinity, while the ratio p over n goes to 0. And so an example would be that p goes with n to the power of 1/3. And then we can show there are no duplicate neurons, and the proof uses a concentration of measure arguments. Now let's suppose we consider such a network or a sequence of networks which gets larger and larger, p equal n to 1/3, and we ask, with such a low rank weight matrix and p equal n to the power 1/3, we can exclude duplicate neurons. Is rate coding still possible? And this will be the second part of the talk. Now let's compare two different networks. The first is a recurrent neural network of rate neurons. So we look at the membrane potential xi of neuron I. It decays. It gets input from other neurons j, and these other neuron j are characterized by their firing rate phi, and they also receive external input. Now this firing rate is a smooth function, a saturating function of the membrane potential x. On the other side, I will look at the spiking neural network. I call the membrane potential now h. It is also decaying. It's a sum over the input of other neurons with weights wij. But now I have spikes, and each spike causes a jump of the membrane potential. And as before, I have an external input current. Now these spikes are generated by an inhomogeneous Poisson process where the probability or the stochastic intensity of spike firing, the momentary intensity, is a function of phi of h, which happens to have the same form as the phi of x on the left-hand side. Now in both cases, I take exactly the same weight matrix. So neuron J is connected to neuron I with this weight here, and this is used on both sides. And with this construction, let us now compare a spiking neural network and a rate-based neural network. Both are recurrent neural networks. Both receive the same external input current. And only one half of the neurons receives the current. The other half do not receive external input. So they are completely driven by the internal connectivity of the network, by the recurrent connectivity. The first half of the network receive input, and this input is also low rank, and we choose it to be the same, and this is a noise process that varies rapidly in time. So this is a very largely fluctuating input current that drives the network, and n half of the neurons are driven directly, the other n half receive input from the recurrent network. And so let's now look at the behavior of these two networks, and we use for both networks the exact same weight matrix. So I can really say this neuron here is the same as this neuron here, except that on the top it's a spiking neural network, at the bottom it's a rate neural network. So this one neuron, let's call it neuron I, has a membrane potential in the spiking network which is hi of t. This very same neuron I in the rate network has a membrane potential called xi of t. And now let's compare these two membrane potentials, and we see they have the same time course, except that the spiking neural network also has occasional spikes. Now we can do this for a different neuron, for example, the one that generates this red trajectory, and we see that it's very similar also for the red neuron. And it's similar for the green neuron, it's similar for the blue neuron, and so forth. Let's try to quantify this. How similar is hi to xi? So in this recurrent part of the neural network, neurons that don't receive external input, we compare the hi with the xi, absolute value. Then we take the average over time, and it's a time-dependent input, so nothing is stationary, as you have seen on the previous slide. And then we ask, on average, what is this distance? And then we do this for different network sizes. The network size is n, and if we change the network size, we can keep p fixed. That would mean we change the ratio alpha, and that's what's plotted here. Now, particularly interesting is this line here, the solid line, which is this ratio p equal n to 1/3. So we know for sure there are no duplicate neurons, and nevertheless, we see that for a big network of 1 million neurons, that this distance measure is just super small. That means the effect of spikes in the spiking network disappears for large networks. There's no need for additional averaging over time or neuron duplicates. Each neuron is different. There are no neuron duplicates. Yet the rate network has the same membrane potential trajectory as the spiking network. The dashed line is a bound for small values of alpha, and you see that the bound gives a very reasonable understanding of the observed behavior. In other words, the spiking neural networks turn into rate neural networks. Let me come back for a moment to the scaling. This is a bound for small alpha. Alpha is the ratio between the number of patterns or the rank and on one side and the number of neurons in the network on the other side. Now, with p equal alpha n, while n goes to infinity, then this distance is always proportional to square root of alpha. So now if I increase p and n at the same rate, alpha is fixed and then this is finite. That means with this kind of scaling, I cannot make the transition from spiking neural networks to rate neural networks. On the other hand, if I take this p equal n to the power of 1/3, then this distance goes down. Let's look at the first scaling where p is alpha n. That means if alpha is fixed, then whether I take p equal 100, 200 or 400, I always land at the same distance. In the second case, if p is equal to n to the power of 1/3, then in fact I move along this solid line and I go down to very, very low values of this distance, and this is the situation where the spiking neural network converges to the rate neural network. So let's go back for a moment what I said earlier. I said that there are different ways to get neural networks without duplicates. We looked at this situation of uncorrelated neurons. P equal alpha n is one of the cases, but P equal alpha n has a problem down here because it doesn't converge. If I take P equal n to 1/3, then I'm fine, I have no duplicates, and moreover, I get a distance that goes down to 0. Another common scaling that you find in the literature is that we have a fixed p, a fixed rank, while n goes to infinity. And in this case, we end up with correlated neurons. There are many duplicates, and we have seen an example of this in the ring model. So it's really a scaling n to 1/3. It could be n to the power 7/8 or 1/4, a scaling that is less than a power of 1, that gives this situation that I have on the one side no duplicates and on the other side I have a distance that goes down to 0. To summarize, rate coding with instantaneous time-dependent rates is possible in a network of spiking neurons, even though not a single pair of neurons is correlated. There are no duplicates. It's a completely heterogeneous population, no spatial averaging, no temporal averaging. Now the reason why it works is this low-rank weight matrix structure that we used for the connectivity. Now these low-rank weight matrices give low-dimensional network input to each neuron, and the neural activity as a result lives in a p-dimensional manifold, and we can choose p equal to 100. So it's not that p is equal to 5 or 6. P can be quite a big number. What's important is that the ratio of p to n, the total number of neurons, is small. So under these conditions, the spiking neural network turns into a rate neural network. So we had this big question, is rate coding meaningful in spiking neurons, even if signals are fast, so that no temporal averaging is possible, if there are no duplicate neurons? And the answer is yes. And we used low rank connectivity. Now low rank, as we said, is related to this maximum number of contributions, big P, and that's called the rank P of the matrix. And as I mentioned, P doesn't have to be 3 or 4. It can be, for example, 100 if I have enough neurons. Now, you may wonder, well, this low rank connectivity, isn't it a strange assumption? You may say neurons have receptive fields and wiring patterns. So is a low rank model that leads to dimensional dynamics realistic at all? And this question has been asked in several reviews. And the answer is all standard models of cortex are dominated by low rank connectivity matrix. So if you think of a decision model, you take it off the shelf, it will be low rank. If you think of associative memory model, it will be low rank. If you think of a neural field model, it's also low rank. Then the second question is, how are low rank networks related to low dimensional dynamics? And this is the very important and influential work of Mastro Giuseppe and Ostojic, who showed that the rank P rate matrix always generates p-dimensional dynamics, and this gives rise to neural manifolds. So, spiking neural networks can transform to rate neural networks without averaging. It's a rather loose condition, this low rank condition, because the rank P can be relatively large, and you can read about this in our paper.